I'd like to introduce Dr. James Connor. He is the Vice Chair of the Department of Neurology at Penn State.
Neurosurgery.
Sorry. And he's going to tell us a little bit about what he does.
Sure. So the DoD grant that we have, the congressional mandated research program was through their Novel Therapeutic Award program. This is a project that we've done jointly with Dr. Amanda Snyder, who's a postdoctoral fellow in our lab. The idea that we had was that there's a fluid that surrounds the brain called cerebrospinal fluid. Most people are familiar with this. If you do a lumbar tap, it's the fluid that's pulled off in the lumbar tap. And so we thought that maybe there is some type of change, maybe a buildup in toxins, loss of nutrients, something that might be gone awry with this fluid. So what Dr. Snyder did was come up with we call an artificial cerebrospinal fluid that she's put together, and it includes a protein that's kind of near and dear to our heart, since we work on iron a lot, a protein called ferritin, which is actually also normally found in the CSF, but we changed it just a little bit so that it normally carries a lot of iron with it. It's the way we move iron around in our body, but in this case we made it iron free when we started, with the idea that it would begin to accumulate some of the iron, as there's some data showing in the ALS individuals as well as in the animal models that there is excessive iron accumulation in around the damaged regions. And the problem with that is it causes oxidative damage. So it really, if it's not causative as far as the disease goes, it certainly accelerates the problem. So with the sort of the artificial CSF in there, with the addition of the ferritin, we were able to show that we could expand the survival time in the animal model of ALS, and the data are very exciting. Actually, Dr. Snyder's at the Society for Neuroscience meeting now, presenting that work, and then we'll present it again later in December at the ALS MND meeting in Milan. She'll present there as well. So it's pretty exciting, and you know, it's always exciting in the animal model, but the question always is, can we take it forward? And what's neat about this and exciting about this is that, you know, the ingredients in the artificial CSF are normally occurring ingredients in the human CSF, so there's nothing in here that isn't already floating around in our body. From a surgical approach standpoint, we think we can use the way we already treat chronic pain with the morphine pumps into the spinal cord. We could take that similar approach into our ALS population. So we really think we have this to a point where, based on the animal data and the sort of the non-pharmacological approach, that we still have to discuss with FDA, and our own IRB what they'll approve and what we can do to go forward. But there's nothing, we're not asking to put something into people that's not already there, and we're not asking for a new medical device. And so we think we're pretty excited about this could move quickly into a clinical trial and see what's and see what's going on, there.
So you're waiting for approval to be able to start a clinical trial, or how does that work?
We're looking for someone to help us, quite frankly, get the funding to do the trial. We're reaching out to a medical device company, reaching out to a number of avenues, with the idea that since they have much more familiarity with going to the FDA, we can certainly work with our IRB here. But we think reaching out to find, because it's still going to be an invasive procedure in the sense of minimally invasive, but it still requires a device and like I said, it's just the morphine, similar to the morphine pump.
Our insulin pump, is that?
Very similar, yeah. So really, as far as those approaches go, it's very minimally invasive surgical approach, and like I said, the fluid that we would be infusing is also it's all naturally occurring ingredients. One thing I didn't mention that we were also pretty excited about is that it's not simply the idea of kind of flushing the system, if you will. You know, the ingredients that we were putting in were really important. So one of the studies that Dr. Snyder did was simply infuse a system, a buffering system, like lactate bringers that we would normally use in the hospital, or a buffered saline solution that we use frequently in the lab. And those animals didn't improve, so it's really not simply just kind of clean out the system a little bit. We really was important to provide this nutriently balanced solution with the addition of the ferritin, which we think was probably providing some trophic support, but probably also sort of chelating the iron.
Chelating, so it's pulling the iron out of the...
It's pulling the iron, and it's already been shown to be effective in an extracellular fashion, in that it can really, it doesn't have to get inside the cell to work. We normally find it in the nervous system. We tend to find it in cells. It's sort of the main iron storage capacity, or iron storage protein for all cells, but it's also very, very high in high concentration in the serum. So what we think it's doing is the word we like to use is a ionophore. So it's a chelator in the sense that it binds iron, but the problem with iron chelators is that they bind iron, and they bind it so tightly and indiscriminately. They don't look and say, oh well, the cell needs that iron to make energy, but this one's causing problems, so we'll bind this one. They just grab both of them. So what our ferritin does is more selectively bind iron, but also once it binds it, it recycles it back in a natural way. So it brings it back to the liver and the spleen.
Okay, so it clears the body, and
it makes the iron available for making red blood cells, which is what we're mostly using our iron for. So again, that differentiates it from a chelator, which would so tightly bind the iron and then probably go hide out in the liver for ever, and so that iron would never be available again.
So your approach is to kind of slow things down. Is that?
We think I think you're right. We're probably, this probably is not a cure. We'd like for it to be, but we have some ideas to push that forward as well. But for right now, what we think is that this would slow down the degenerative process, slow down the progression. Exactly.
Can you talk a little bit more about how the iron, the collection of iron, affects someone with ALS?
Sure. It's always, when we talk about oxidative damage, and it sounds bad, and we try and think, but I always tell our students, the best example of a oxidative modification to something is the egg white. So you crack open an egg, and that the non-yolk part, when it turns white, that's an oxidative reaction. So if you sort of envision your cells in your body being that nice fluid, sort of part of the egg, then it becomes congealed and hard, and so the proteins that should be moving around in the membranes in your cell, they get stuck, and they can't go anywhere. So there's a couple of things that happen. They get stuck, so they don't signal the way they should. So the nerve cell is not getting the right kind of information, and when a nerve cell doesn't get the right kind of information, it dies. That's programmed in. If you're not doing your job, you die, because there's hopefully other nerve cells around that'll for whatever happened to you, that they'll pick up the pace. But unfortunately, in the sense of ALS and motor neurons, there's a finite number, and so as they begin to die and the disease expands, we end up having the problems associated with ALS. So that's really. And so what iron does is promote that oxidative process. So one of the fun things we've been working on iron and challenging is it's always about the balance. We absolutely need iron to generate energy. So the very chemical capacity of iron to interact with oxygen that allows us to make ATP and energy is the same problem that causes us to have oxidative stress. So it's that ability of iron to accept and donate electrons, to sort of get into the chemistry of it, which is like I say, absolutely essential for us to use oxygen to make energy. At the same time, if the reaction goes in the other direction, it turns the oxygen into these free radicals that cause this oxidative damage, and so that's the problem then, that, as I said, that promotes the cell death. So even if it's not a causative factor in the disease, and it may not be, but what it certainly does is promote the progression of the disease.
What are you talking about as far as a timeline? Any ALS patient would ask you this.
Right, yeah, no, it's a great and fair question, and my answer always is, I wish it was yesterday. We're talking to as many people as we can, seeking funding from as many organizations as we can, and bottom line is, I think, once we find a partner, we're from standpoint of our hospital, our neurosurgeons are ready to go. Dr. Zach Simmons is our director of the ALS program, and as everybody knows, a wonderful guy.
Wonderful.
And physician. He thinks we're thinking about this the right way, so everybody here is ready to go. It's a matter
of the
funding and approvals, and the approvals again because of, as you know, the problems with not having any really good treatments for ALS out there. It's not like we have to look and say, well, we've got something extending lifespan by 5 years, so in order to go into the clinic, we've got to be at least better than 5 years. We don't, and sadly the bar is set kind of low here. So if we can, we have a treatment, and as you know, phase 1 trials are basically set up to make sure we're not going to do any damage, and so the first trial would be in individuals who are not doing very well, and probably fail medical management at that point, just to see if we can come in and show some improvement. And so, another study that we've done, which will probably link into this, is one of our MD PhD students named Bill Sue has just had a paper come out. We're looking at biomarkers in the CSF, again, of ALS, CSF and blood. And so he's published a very nice paper now that looks like we found a profile of proteins that have pretty significant predictive value in terms of the disease progression.
Okay.
So one of the things that we would do as a very early step in their study is, as we're infusing our hopefully nutrient healthy solution into the CSF, we would be monitoring changes in these protein profiles that, as it looks like that we can predict the patient outcome. And so to see if those are changing, so we'd actually be able to monitor and see what we're doing during the course of the study. So,
protein is something that has come up so many times today. Can you talk a little bit about the protein?
Sure. Yeah. I think the mistake that has been in our, we want a biomarker. The ALS is not that difficult to diagnose. There's some ALS mimics, but what Dr. Simmons, what our clinicians need help with is when the patient comes in and they're pretty sure of their diagnosis, but they don't know if it's going to be 2 years, 5 years, 10 years, and of course the patient wants to know, the physician wants to know what treatments are available. So the proteins that we can look for, these biomarkers, what we all want is something to say, well, when this goes up or this goes down, now this is, if you need help in the diagnosis, this is help in the diagnosis. But as we said in ALS, more importantly is saying, if this is over this level, this is not good. We're worried that this is the sort of the 2-year sentence, and we need to be as aggressive we have, or gee, this protein is really low, so we're going to watch, but when we see this protein low, then hopefully this is the 5-10 year plan with the disease progression. But as I started to say, I think where the field has suffered, and not just in the ALS field, but in general with biomarkers, is trying to find that one protein, the one magic bullet. It's almost assuredly not going to be one. It's going to be a profile, and that's what we've been doing, what Bill Sue's been doing, is we have a profile of about 12 proteins, and so you not only look at those 12 proteins, but look at them in relationship to each other. So it's not just this one's high, and these three are high and these three are low, but when these are in some relationship to each other, and sometimes the relationship between what's in the CSF and what's in the serum turn out also to be very informative. And so that's when we say about this protein biomarker, that's what we're looking for, whether these are what we call inflammatory cytokines that we know go up when an individual's sick or cells are dying. So those are the different, and the other nice thing about that is because all of these proteins have sort of known functions, what we can look at and say, well, gee, if that's up, then this one's down, that must mean the certain pathway is being activated or going on or being deactivated. So it also gives us the opportunity to have new drug targets and say, well, if the inflammatory, we don't have to worry about anything until the inflammatory profile goes up. And so when the inflammatory profile goes up, that's the time to treat. When that's low, our treatment strategies may not be helpful because they're targeting something that's not bad at this particular moment. So I think that's the other part of where we would benefit from this type of biomarker research. What people would be paying for is the device, the pharmaceutical time to put the Pharmaceutical Services. It's not a drug, but somebody still has to put the solution together in a sterile environment because it's good. So we have particular setups in our pharmaceutical department that would put these together, and then the OR time to put in the device.
What are some misconceptions that people have about ALS and research and possibly research in general?
Seems like we should be able to understand it better. It's a really hard thing, I think, to talk to a patient, talk to somebody about ALS and say, well, here's what's going on. Say, if we know what's going on, why can't we fix that? And so I think projects, I think that's, so I think that sort of the misconception is that, and it's hard because I agree with them actually that pardon me, that why don't we fix this? And so you start. I think the misconception for me, the biggest misconception is we're going to find a gene, and gene causes all motor neuron degenerative disease. I mean, we already know. We found a number of genes, and we spent lots and lots of money on lots of genetic studies. And what we know is, there's multiple genes, and none of them account for a whole lot of the disease. So I think sort of like the biomarker idea, where we think we're going to, I should say, we think there are studies that say or groups that think or looking for that one protein that's going to change and it's going to tell us everything. We pretty much know that's not the case. I think we pretty much know from genetic studies that's not the case either in ALS. And the other part of that story is that even if we did find a gene, the gene isn't the disease. The gene is the mutation that puts the disease in progress. We always say, even the SOD1 mutation, which
It's
is
the light switch.
It's the light switch, but we need to learn where to treat, we need to learn, you know, Huntington's disease.
Learn how to
Yeah.
turn off that light
Yeah,
switch.
yeah, you know, I mean, Huntington's disease is, you know, an example. You know, we know the gene, we know the protein, we know the disease progression, you know, but we're still not very good at treating it. You know, the different mutations that come out that people think are involved in Parkinson's, the same thing is that, you know, we don't have people with a mutation that are born with Parkinson's disease. You don't get Parkinson's until you live long enough to get it. And then ALS is very similar. So, you know, even if there is a gene or a set of genes that finally get defined, you say, well, you know, why are some people getting ALS at, you know, in their 20s, 30s, 40s? You know, if there is a gene, it is serving as the trigger, but it might serve as the trigger, but what are all these other things that are impinging upon there to actually, you know, manifest as a disease? So I think those are, you know, we're not going to, as I said, identify a gene and then the following week have the cure for ALS. I think that's one of the misconceptions, and unfortunately, I think a lot of resources are going into learning that it's not. You know, I mean, you can't say you know, because it's all knowledge and it's all contributing. Unfortunately, we're contributing information that says it's not this, and you know, that's as important. You know, from a scientific standpoint, that's as important as learning what it is. But from a patient standpoint, it's they're happy that we're ruling out things.
Yeah.
But if you're a patient with ALS, you want us to be taking some more directed action.
What can ALS patients, their families, friends, everybody, what can they do to help you to do this?
I mean, I think we're continue to donate to the association, working with you guys in the association to put in our proposals and continue to get support. That's unfortunately where the really new ideas come from are really going to be is more of a grassroots movement of support. So it's really our, as you say, our patients and our families and friends of patients that are going to help us move forward in something that's out of the box. Really come from, I think, again, the grassroots efforts. It has to be a disease that's of interest to the military, and as you know, I mean, ALS has prevalence in the military. So I think, again, getting a hold of their congressman. I know Congressman Dent that we've met has shown a lot of interest and support for the program. So I think locally here through Congressman Dent, but in general throughout everybody that the ALS Association reaches, to talk to their congressmen that continue to support to those programs at a level that will allow us to get the work done.
It sounds to me like you sound very hopeful about this procedure.
We are. We really are. I mean, the animal data tell us we're thinking about this the right way. And again, we're not saying with you it's going to be years of development and attempts to get approval, because it's we can go down to the pharmacy right now and make this. I mean, literally right now. And the device guy was walking around. We grabbed him, and our neurosurgeons totally on board with this, and so we could, if everything were in place, we could do our first patient tomorrow.